You know, inside every single one of your cells, there's an instruction manual that's 3 billion letters long. It's the code that literally builds you. But what happens when there's a typo in that code? Well, today, we're going to trace that incredible journey from a line of DNA to a working protein and see what happens when the code goes wrong.
It sounds kind of crazy, right? How can one single mistake, just one letter swapped out of 3 billion, be the difference between perfect health and a lifelong disease? That's the huge question we're going to unpack right now.
All right, here's our game plan. First, we'll look at why DNA is the ultimate instruction manual. Then we'll get into the step-by-step of how your body actually reads that manual. From there, we'll see what happens when the code breaks, compare the different kinds of typos, and finally, see what this all means in the real world.
Okay, let's kick things off with the big picture. Just think of your DNA as this massive, super detailed blueprint for building and running your entire body.
Now, this blueprint is broken up into chapters, and we call those chapters genes. Each gene has one job, to provide the instructions for building one specific protein. And proteins, they're the real workhorses of our cells. They do pretty much everything. Take insulin, for example. It's a protein that's absolutely vital for managing sugar in your blood. If the instructions for insulin are messed up, your body can't make it right, and that can lead to diseases like diabetes.
So, how does a cell actually take this blueprint and build something with it? Well, it's this really elegant two-step process. It's kind of like a factory assembly line. Let's break down exactly how it works.
The whole thing starts with step 1, which is called transcription. This is where a copy of the instructions gets made. And then comes step 2, translation, where that copy is used to build the final product. It all begins in the cell's main office, the nucleus, and finishes out on the factory floor at a little machine called the ribosome.
Okay, so let's follow the recipe for a piece of the human insulin gene. In transcription, the DNA double helix unwinds just a tiny bit. Then, an enzyme comes in and makes a temporary copy. We call this copy messenger RNA, or just mRNA. It does this by matching up bases to the DNA strand. Here's the key rule. C always pairs with G, and A in the DNA pairs with a letter called U in the RNA. So that's why you see every T in the original DNA code becomes a U in the RNA copy. This mRNA is now the working blueprint ready to head out of the nucleus.
So how does the cell make sense of this long string of letters? Here's the really cool part. It doesn't read them one by one. It reads them in groups of 3, like little 3-letter words. Each of these words is called a codon, and every codon is an instruction for one specific building block, which is called an amino acid.
And that brings us to step 2, translation. The mRNA message leaves the nucleus and finds a ribosome. The ribosome is like a little code-reading machine. It latches on and starts zipping along the mRNA, reading each 3-letter codon. For every codon it reads, it grabs the right amino acid and adds it to a growing chain. So it reads GGU and adds glycine. Then it hits AUC and adds isoleucine, and so on, all the way down the line, building this chain of amino acids. And once it's done, that chain folds up into a complex working protein. The bottom line is this. The DNA sequence dictates the amino acid sequence, and that sequence dictates the protein's final shape and its job.
Now, that's what happens when everything goes perfectly. But what about when there's a mistake in that original DNA blueprint? Well, these mistakes are what we call genetic mutations. Just think of them as typos in the instruction manual.
One of the most common types of typos is a point mutation. This is where just a single letter, a single base in the DNA gets changed. Maybe a C accidentally becomes a T. And just like we saw with the insulin example, that tiny, tiny change can cause the wrong amino acid to get put in the chain, which can mess up the protein's shape and stop it from doing its job.
But here's where it gets interesting. Not all typos have the same effect. There's a world of difference between changing one letter and, say, deleting one completely. So let's compare the two big types of mutations.
Before I just give you the answer, think about it for a second. One typo just swaps a letter. The other one adds or deletes a letter, which shifts the whole rest of the sentence. Which one sounds like it would cause more chaos?
So here's the deal. A point mutation is like changing one letter in a sentence. The sentence, the cat sat, might become the bat sat. The meaning changes, for sure, but the rest of the sentence is still perfectly readable. But a frame shift mutation, oh, that's way more destructive. It completely scrambles the reading frame because the ribosome is still reading in groups of 3.
If you delete the E from the cat sat, the whole thing becomes gibberish, thur hat sit. Every single codon from that point on is totally wrong, which usually results in a completely useless protein.
But not every typo is a disaster. Sometimes a point mutation can be silent. This happens because the genetic code has some built-in redundancy. A couple of different three-letter codons can actually code for the exact same amino acid. So the DNA might change, but the protein ends up being exactly the same. It's kind of like spelling the word gray with an A or an E. It looks different, but the meaning is identical.
Okay, so let's bring all this theory into the real world. A lot of really serious genetic diseases are the direct result of these tiny typos we've been talking about.
Let's start by looking at the gene for beta-globin. It's a key protein in your red blood cells. Here's a little piece of the normal DNA, and you can see the amino acids it codes for. Pay attention to that fourth one in the list, glutamic acid.
And here's the change that causes sickle cell anemia. It's a single point mutation. One T in the DNA has been swapped for an A. And what's the result? That one little change swaps out glutamic acid for a totally different amino acid, valine. It seems so minor, but that one single swap causes the whole hemoglobin protein to fold up wrong, which creates those sickle-shaped red blood cells that just can't do their job properly.
Cystic fibrosis, on the other hand, is a textbook example of just how devastating a frameshift mutation can be. The disease comes from a faulty protein that's supposed to move chloride around, and very often, the mutation is the deletion of just a couple of DNA bases. But as we saw, that shifts the entire reading frame, turning the rest of the instructions into total nonsense and producing a completely broken protein.
So just to sum it all up, we have point mutations, which can cause diseases like sickle cell anemia. We have frameshift mutations, which are often much more severe and lead to conditions like cystic fibrosis. And there are even other types, like a nonsense mutation, which changes a codon into an early stop signal, cutting protein production short. That's what happens in some forms of muscular dystrophy.
In the end, it really all boils down to this one fundamental idea. The order of the letters in our DNA determines the shape of our proteins, and the shape of a protein determines its function. A tiny change to the code can cause a massive change in that function.
And that leaves us with one last thing to think about. Our genetic code is this incredibly powerful, unbelievably complex blueprint, but at the same time, it's so fragile. Knowing that just one letter out of 3 billion can change a person's entire life, well, it really makes you stop and appreciate the incredible, delicate dance of biology that's happening inside all of us every single second.